We're going to begin this video discussing post-transcriptional mRNA processes. In the last video, we talked about transcription and how we got to pre-mRNA from DNA. Today we're going to talk about what happens after transcription. mRNA must be processed for quality control. These include stamps that approve the sequences. These additions are added at the beginning and the ends of the pre-mRNA. The 5' is a methylated guanine attached to the 5' end of all mRNA. All phosphates remain, and this allows for recognition of a genuine 5' end on mRNA. A poly-A tail is a run of adenine bases added to the 3' ends of mRNA and marks a genuine 3' end of mRNA. All enzymes for these modifications are carried along transcription and are used when needed. These marks can now leave the nucleus safely and be labeled as mature mRNA. Proteins interact with both ends at the same time. This means that the 5' end will interact with the 3' poly-A tail and create a circular mRNA strand. This double checks for genuine caps. They must be able to interact with one another. mRNAs are chopped up and glued back together before translation to remove non-coding sequences. This is known as transcriptional splicing. There are introns that are intervening sequences that do not code for anything. Introns can be in high numbers and can be similar sequences, but they aren't used for anything. Exons are the functional coding sequences that we must keep. RNA polymerase has already transcribed the introns and the exons together. In order to splice these introns, it is done with a large protein complex called a spliceosome. This spliceosome splices and removes the introns and ligates the exons. The 5' ends of introns that have been spliced create a lariat or a loop to its 3' end of its intron. It will then be chopped and removed for the exons to be ligated together. Although some exons may not be adjacent, they can still be placed together. This results in the creation of new sequences and new proteins. If you see here, you can see that when leaving transcription, pre-mRNA begins to become mature mRNA because it is now containing a 5' cap, and the poly-A tail is going to be placed at the 3' end. You can see here that there is the 5' cap and a poly-A tail, but that does not make a finished mRNA product. There must be the removal of the introns. You have introns here and introns here. These will be spliced out, and this will be a remaining protein coding sequence with just exons. It also contains the 5' cap and the poly-A tail 3'. This is now a mature mRNA and can now leave the cell in order to go through translation. Translation. Ribosomes are used to make proteins. They are used to coordinate and assemble the proper complexes in order to create amino acids. There are two subunits, the largest subunit and the small subunit. They are naturally found with the large subunit placed on top of the small subunit. mRNA will initiate translation when mRNA is sandwiched in between the subunits. The large subunit is a complex that contains 49 proteins and 3 RNA molecules. The small subunit are made up of 33 proteins and 1 RNA complex. The larger subunit catalyzes the peptide bonds between amino acids and allows for the creation of proteins. There are 3 voids found in the large subunit that are for tRNA binding. There is the A site, which is the amino site and only carries 1 amino acid. The P site are for the peptidyl tRNAs, which hold peptide bonds between amino acids, or peptide chains. And the E site is known as the exit site. There are several factors needed for translation initiation, such as the initiation factors. These bind to the 5' cap of mRNA and the subunits. They bring the proper translation factors together. tRNAs are known as transfer RNAs. They are also known as true translators. They are adapter molecules that interact with both codons and amino acids. tRNAs have codons that are known as anticodons. These codons correspond to the codons found on mRNA, such as a start codon. These nucleotides are base pairing and are antiparallel to the anticodons. On the other end of tRNAs are the amino acids that correlate with that anticodon. Hence, that codon found on mRNA. tRNA are the only molecules that work directly in between codons and amino acid language. In order to begin translation, there is a need for the initiator, containing that start codon, that AUG codon, or that start anticodon, CAU. Once the initiator tRNA is bound to the small subunit, the large subunit is called upon. This will land and leave the initiator tRNA in the peptidyl site. As you recall, the peptidyl site must have peptide chains, and this only has 1 amino acid attached. This is an exception to the rule because soon after, a new tRNA will begin entering the A site with the proper amino acid and anticodon that will bond with the mRNA codon. The amino acid will create a chain with the peptide bond in the P site, and that tRNA must now move to the P site. That start tRNA is now at the E site and may leave the cell, the subunit. In order for this movement to happen, the ribosomes must translocate. The ribosomes shift forward with each codon and will move the A site and P site to fit the appropriate tRNA. Only 2 RNAs are allowed in the ribosome at a time. So the tRNA moved to the E site will leave, and a new A site tRNA will enter. In other words, mRNA is pulled through the ribosomes and codons are read through site to site and moved site to site. tRNA form anticodon and codon interactions, and amino acid chains will follow. Translation termination is signaled by stop codons and release a factor protein when it enters the A site that will bond to the codon. These release factors sever the bond of the polypeptides found in the P site and will translocate once again to release that tRNA and shut down this process completely. If we look at this image here, you can see how beautifully this is done. You can see here we have this incoming tRNA with the corresponding anticodon to this codon region on the mRNA. Attached to the tRNA is that corresponding amino acid. Once this comes in to the A site, this is transferred to the P site with the following, connecting this chain of amino acids. The amino acid is released from the tRNA, and this tRNA is released from the ribosome units. This may look more complex, but it shows everything from transcription through translation. If we start up here, we have transcription, where you have your pre-mRNA. Then you have mRNA processing, which add the poly A tails and the caps. Now mRNA is ready to leave and enter the small subunit, where it can enter the A site, be moved to the P site, add that amino acid to the chain, and be released out of the ribosome once that chain is created.